everyone, and Happy New Year. Some of you haven't yet, so wish you a lovely 2026. It's my huge pleasure to have here today with us Jessie, so Dr. Mosso. In fact, why it's such a huge pleasure? Just because Jessie was with us in the past. Jessie was one of my PhD students. Now she's not anymore. And we had a lot of fun together. At least I hope we worked a lot together. And Jessie was the one who implemented the diffusion weighted spectroscopy in our group. I remember that she started in 2019, and it was amazing to work with her. So this is why she's also here today. In fact, we miss her a lot. So today we invited her. She was traveling in Europe for the new year. And we invited her with us to have a little bit of insights of her work, what she's doing in New York. So now Jessie is a postdoc in New York in a very well-known lab of Novikov, of Professor Novikov, and also Fiermans. And she's working a lot on diffusion. So now she works with experts, with real experts in diffusion. And she's switched a little bit from preclinical diffusion weighted spectroscopy to so she will present a little bit of what she's doing, and thank you very much for being here with us, and I'm looking forward to see what you'll be saying.
Yeah. Thank you very much for the invitation, and yeah, it's a great pleasure to be here to see so many of you that I haven't seen in a while, so it's thank you. So I'll be presenting the work I've been doing for the past year at NYU, so it's diffusion weighted MR spectroscopy and diffusion weighted MRI in human brain. So I shifted from preclinical to human brain. So first for the introduction, so this is a bit the picture we are trying to do in diffusion MRI. So we are trying to access from MRI image. I don't know if you see my mouse here. So from MRI image, we are trying to access the physical properties of a small voxel here, so at the microscopy level. So this is our reality. It's a micrometer scale, and this is really what we are trying to access with diffusion MRI. To do this, we use what we call biophysical modeling, which is this part. So we describe the voxel in terms of different compartments. So you may know that we use, for example, sticks to describe axon fibers. We have extracellular space, free water, etc. So these are the compartments we use in our biophysical modeling, and then these compartments create what we call a kernel that is oriented in a specific manner in the diffusion MRI voxel. And the beauty of diffusion MRI is that this is also the micro scale, so it's a bit surprising because you may think that the MRI voxel is at the millimeter scale. But because we have this very nice property of diffusion, that the diffusion length scales with square root of dt, you actually can probe with t, so experimentally accessible diffusion times. You can actually probe lengths that are in the scale of the micrometer as well. So it's really nice property of diffusion in general. And so a bit more about this biophysical modeling, because this is the clue of my work now. So we describe the diffusion MRI signal as a spherical convolution between what we call a kernel, which is the K here, and what we call orientation distribution function, the P, which is how the kernel is oriented in space. So if you look at this picture again, the kernel really says about the microstructure. So this is something that you will usually find in literature of diffusion MRI. We are trying to look at the microstructure features of the voxel, and this is the very rich part of our signal. So it's specific to different tissue types. So you have white matter model that's different from gray matter model. And it's also specific to distinct biological processes, so you can probe neuroinflammation, bleeding, so different sorts of. So it's very rich information from the kernel part. And the other part of the diffusion MRI signal is how is this kernel oriented in space within the voxel, so what we call the ODF here, that I will use this term all along the slides. So it's really about the spatial distribution of this kernel, and this is what you may know as the basis of tractography. So when you see this tracking the fibers along in the brain, for example, this is based on this measurement of this P function here. Why am I saying this? Is that we are trying to extract the kernel. It is when we are doing this, it's a very difficult problem. So it's a ill-posed problem. We have many solutions that bring the same kernel, and it's also generally harder to extract the kernel than this P function here, so the ODF. So people have used several strategies to be able to still fit this kernel and alleviate fit degeneracy. So most of models that you know here, these are white matter models. They use constraints, so either they fix some diffusivities in the model, or they assume a certain hierarchy of diffusivity, or they use machine learning, or so the different techniques have been used to help fitting and retrieving the kernel features. However, still, when we are talking about diffusion MRI models, we are still talking about one kernel, which means one type of sticks, and in general, it means it represents only axons. And the question that we may ask is, even in the human brain, what about glial cells? So if we think about just one kernel, we think about neurons mostly, but what about glial cells? So the main statement is that when you look at diffusion MRI biophysical models of white matter, glial cells are completely ignored for several reasons that we may understand. The volume fraction of glial cells is very small, so you may not want to model them, or you may not need to model them. There have been also conflicting results regarding the need to include them and their impact on the results for extracting kernel features. And diffusion MRI doesn't have the cell specificity that other modalities can have, so it's difficult to separate glia from neurons. However, glial cells are still present in human white matter. You have oligodendrocytes, part of the myelin sheath. You have fibrous astrocytes that also go along the fiber tracks. And there is a growing evidence for their role in neurological disorders. So we may also want to look at them just because they might provide very important biological information. And here comes diffusion MRS, which is the second modality I'm working with. And this technique, as you know, allows to probe glial cells because we benefit from cell specificity of metabolites. And some work has been done already in this field. So people have looked at glial pathologies in human brain, and mostly focusing on low B value protocols, but they have been able to probe a specific alteration of glial metabolite diffusion properties. And there has been also extensive animal work on more high B values, so more complex protocol, diffusion time dependence. This was pioneered by the group of Julien Valette, and then also Cristina and our work following. And there has been evidence for different glial microstructure, so that diffusion MRS could probe this microstructure. And more recently, there was the work of André Doring that's here, who extended this in human on the connectome scanner with very strong gradients. But still, in the general field, we are still missing studies in human white matter and to try to characterize this microstructure of glial cells. So these were the main questions of my work so far. Can diffusion MRS probe the glial compartment in human white matter? And if so, do the glial and neuronal metabolites probe the same kernel and the same ODF? So again, the orientation of the kernel within the box. So this is going to be the two parts of my talk. We addressed both, so the ODF and the kernel, and compared what we, in this large white matter voxel that we work on, how the glial and neuronal metabolites differ in what they represent of the ODF and the kernel. And finally, I will present you our very first result on our new Connectome X scanner. So it's you may have heard about it. It's very strong strong gradient 3D scanner that was developed by Siemens very recently. So before I dig into part one, there is one aspect that should be addressed when we discuss all these biophysical models and we discuss about sticks. It is so depending on the acquisition protocol, you will be sensitive to a different aspect of the stick, which is the basic structure of all our models. And this depends on the diffusion time. So you can see here the diffusion times corresponding to a certain diffusion length. And while you increase the diffusion time, you see here you homogenize the windows through which you look at the stick. So at the short diffusion time, you will see a very complex structure. But as you increase the diffusion time, you see something that approaches a one-dimensional, very straight, zero radius stick. And this is very important because depending on where you are with your diffusion time, you see a different type of structure, and you cannot assume that the stick model is valid if you're at very short diffusion time, for example. So this is usually in this late regime here at long diffusion time, we usually assume that diffusion is Gaussian. However, if you look at the short diffusion time here, we usually say that diffusion is known to be non-Gaussian, which has several implications. So the first one is that you cannot just describe the linearity part of the diffusion signal. So it's not just about diffusion, it's also about kurtosis, so higher order terms that enter the equation, so deviation from linearity. And all this diffusion, kurtosis, what we call cumulants, they have time dependence. So they depend on which time at which time we are doing the acquisition. And this is a concept that was introduced by Dmitry Novikov, my PI, which is the concept of coarse graining. So you have incomplete coarse graining in this region, while the coarse graining is complete in this region, and you can assume Gaussian diffusion. Why am I mentioning this? Is that we are in between these two regimes for my work. So we first looked at the ODF, so the orientation in space, and then we worked at very long diffusion time. So in that case, we assume that diffusion is purely Gaussian, and we really look at just the diffusion coefficient, no time dependence, and this will give information about, again, the orientation. And in the second part of the talk, I will be really working at this in this regime here, where we are at shorter diffusion times, and there we cannot assume the model of the stick to be valid anymore, and we will access specifically the details of how they deviate from how the neurons, or what we model as neuron, deviate from the stick information here. Okay, so this is now I'm digging into the first part. I forgot to say, if you have any question in the middle, please ask, interrupt any time, or at the end, it's all good. So I will start with a bit of theory, how we describe how we reach this ODF, how we can extract this function. So I told you that we described the diffusion MRI signal as a convolution between the kernel and the ODF. And what we are looking in this part is really how do I access this ODF, which is really the orientation in space. The trick that we use in diffusion MRI often when we want to extract one of these is that we can project. We can write, let's say, this equation in another basis, which we call the spherical harmonic basis. It's a basis of spherical functions, which is convenient for some reason in this case. So we can just expand the signal in another basis. And the coefficients we will be looking at are these PLMs here, which describe the P, characterize the ODF. So this is really the coefficients we are trying to extract. And when I say it's a convenient basis, it's because here we have a convolution that transforms, that converts into a product, because this operation is a bit like a Fourier transform on sphere, so it's just technical details. But overall, we use this equation, which is now a simple product between a kernel and ODF parameters that we can simply solve. If, for example, if we know the kernel, we can just solve this equation and get the PLM. So we have found a convenient basis to convert the convolution into a product. So it's just the message here. Okay, so this is just a technical slide. If for people who are experts here, this is how so we are still looking at this PLM. To do that, we need first to find the kernel. So the kernel, we will need to, for metabolites, we will use spherical mean to derive, to find F and intrinstic diffusivity. We also have access to water, so I will show you later why we have access to water, because of metabolite cycling. So in the same diffusion MRI sequence, we also have access to water. So we have a different way of finding the kernel for water. And then we do spherical deconvolution. So when we have this information about the kernel, we just solve this equation and we find the PLMs with L max to 4. These are really technical details. And at the experimental level, what it means for diffusion MRS is that we will acquire a high B shell with a lot of direction. So this is usually what is done in diffusion MRI. For us, it's very long because each direction is a spectrum. So we have to accumulate a lot of averages. So this experimental protocol is quite horrible for people laying in the scanner. But this was our idea, really, to mimic the way that diffusion MRI acquires a protocol for tractography, for example. So this is what we did. We have 3 volunteers for this protocol. The acquisition time is an hour and a half. This is the position of the voxel. So we tried to find the biggest voxel possible in the white matter region. So it was a bit of a challenge. So you see here we have a region here that is at the intersection of the CST and the SLF. It has crossing fibers. So there is the main fibers going feet to head, and then there is a anterior posterior fiber as well. So it's not free of crossing, but this is the biggest we could find. And then for this protocol, we have this we have a fixed diffusion time, so a long diffusion time, also a long TE. You may notice the TE here is very long. And we have a low B shell, which we use to fit the diffusion tensor, and we have a high B shell, so B8 with 21 direction, really to fit the ODF. The processing is done in standard fashion following the consensus paper. The tricky aspect was the dealing of the macromolecules. So we went to long TE in order to be able to afford not having any macromolecules in the base set. Diffusion MRI was also acquired with in this protocol. So we have very comprehensive diffusion MRI protocol with free gradient waveform, multi-shell. So we could really fit everything we wanted in a very short time. This is the spectral quality we get. This is one of my only spectroscopy slides, so I hope you enjoy it. This is a spectral quality we got for all the 3 different shells. Because of the long TE, we could quantify only 3 major metabolites, so total Na, total creatine, total choline. So it's really, and we use in this full talk total Na as a neuronal marker and total choline as a glial marker. Okay, so the first thing we did in this to still look at the ODF was to look at first the shape of the diffusion tensor. So we focused on the low B shell here only, so B 1.4, and we compared the 3D shape of the tensor for water and different metabolites. So here, what we can see overall, so we compared, this is the 3D representation. It's not very clear maybe, but V1 is the first eigenvector, and lambda 1 here are the magnitude of the eigenvalues. So overall, what we can see from this side is that water metabolites do not really differ in terms of either magnitude or direction of the main eigenvector, and that the FA is also very similar between water and metabolites. You see 3 dots on my individual volunteers here. So conclusion from this low B shell results is that you have similar tensor for water, neuronal, and glial metabolites. So yeah, from this shell we cannot say much. Then we went to find this ODF, so really with the high B shell, the multi-direction, and here is maybe our most striking results for this part, is that you see that, so this is what we call the glyph. So in diffusion MRI, they plot this glyph, but for every voxel. Here we just plot one glyph for the entire voxel for water and the different metabolites. And you see strikingly that the shape of this glyph for water, here the 2 versions of water from MRS and MRI, and for Na are very similar. And this, you see my 3 volunteers here. So this agreement between Na and water is very striking. And if you look at other metabolites like total choline or total creatine, the shape of this glyph seem to differ. So this is really what we observed here. Just a comment that this second volunteer had the contamination of gray matter. So you may wonder why these 2 look similar, but not this one has a long fiber on another direction. So this might be that just the voxel is placed in a wrong position, or either due to motion or just wrong programming of the prescription of the voxel. And yeah, just as I mentioned before, we are looking at this PLM. So these PLMs are really the parameters that characterize this glyph that I was showing in the slide before. And we were trying to see here how much they correlate between water and metabolites. And just you see that for water, so PLMs for water found with diffusion MRIs correlate with the ones from diffusion MRI. Same for NA, but they do not really correlate for choline. So we have we can say that beyond DTI, so when we really do this ODF, we have similar ODF for water and Na, but not for choline. So this is really the main result of this part, that even if we look at this huge voxel of diffusion MRS, we can probe a glyph, ODF, that is seems to be the same for water and NA and not for choline. So we may be able to see the overall distribution of glial cells, which is different from the ones of neuronal, which we expect NA to mimic water, because they represent really the neurons and the axons in white matter. Okay, so then I will go to part 2. I don't know if you have questions on this already, or yeah.
Mm-hmm. Yeah. Would this be interesting to go to another voxel, which is in gray matter, and see if you get this? You will not get the same things if your theory holds.
Yeah. So Louise asked me this before already. So I think so. I asked my colleague who's doing tractography. He says in gray matter, because you have the fibers you have no major alignment. So I think tractography in gray matter is really it's not. Yeah,
but
that's
the microstructure model will now work in base currently from the white matter microstructure model.
And you have to adapt it for
And
you could try it like this and then adapt it to see what it might validate what you see, or might bring addition. I don't know.
Yeah, we had this discussion, and it's maybe something we could try to see if it gives something, and that's the glyph of gray matter. Next step. Yeah.
A question too.
Iliana as well. Yeah.
If I may. Thank you for explaining it very nicely, and I finally understand the abstract I'm an author on. But at the very end, I was still getting confused again about the glyphs. I mean, you say essentially the DTI part, the glial compartment would be similar to the neuronal compartment. But if you go to the glyph, it's different. But still, the sort of DTI parts of it are still similar. So what is the interpretation? You have parts of the glia which are still aligned along the neurons, but you don't have crossing fiber, or I'm confused again.
No, I think the argument was saying here at the low B shell, I think we might not be sensitive to differences. They probably exist here as well, because if we claim they exist, they should exist here, but we are not sensitive to them at this low B shell. So this 1.4 as well is a bit too low for metabolites to probe. This could be value for DTI for water, but not so good for metabolites. So we are really in a regime where it might be just difficult to see it. And the fact that
but the lower coefficients in your high shell version are still aligned. So some compartment is still identical in both glial and nerves. Is that correct? I mean, maybe you have strong dots there with the P2.
So here I didn't describe it, but the round dots are the P2 M's, and so we have 15 coefficients to describe this entire glyph, and these are the lower L2 and then L4. L4 are more noisy than L2, so it's like but this is done on a high B shell, so you cannot just compare the one before, which was done at a low B shell. This is really the tensor, and this, even if it's P2, is you think as the first order, this is still acquired on a high B shell where you suppress extracellular waters. I mean, so it's it's not a straightforward comparison, even if here the P2 are more aligned. Yeah, but this is still a valid question whether to know that to be sure that the deviation we see here comes mostly from L4, so the P4Ms, and they are more noisy. So it still remains the question whether are we just measuring noise, and this is just an effect of noise, and or is this really an effect, and we have a true deviation. Yeah, remains the question.
Thank you.
Yeah, Iliana.
Yeah, thanks. I don't want to stall your second part of your seminar too much. I'll try to be quick. This is really really very cool and fascinating work. I was just wondering, in this long diffusion time regime for the glial cells, how fair is it to use a kernel that's a stick? Like, is it really fair to assume that's what they behave and look like? Because somehow the soma is bulkier and they have a lot of branching, and the segment length is maybe shorter than in neurons. So is this kernel that you chose for the deconvolution suitable?
Yes, that's a good question. I think our argument was that in white matter, even if you have glial cells, you have mostly the fibers of glial cells, which the fibers, there's literature saying the fibers, astrocyte fibers would align along the neuronal fiber tracts. And the soma, I'm not sure I would need to know what's the fraction of soma in white, of glial soma in white matter, which this I don't know, and I'm still trying to find a good literature reference, even for just pure volume fraction of glial cells in white matter, and what kind of glial cells are we talking about. So yeah, I'm still, that's the valid question. If you have a non-negligible fraction of soma, yes, this is not accounted for in the kernel. But yeah, if you have any idea of a good literature reference for contribution of glia in white matter, that would be.
I mean, we have some ballpark figures from some literature. I don't know what they're worth, but we can share them with you.
Yeah. Thank you. Yeah. Okay, so I'll go to now the second part. So once again, so the first part was about this orientation. Second part is about the kernel. So it's what we call the difficult part, because extracting these features is usually more difficult. So here also a brief introduction about the theory. So I told you initially that we are now in a regime where we have non-Gaussian diffusion. So we will work at shorter diffusion time. So now we need to account for different things like higher order cumulants, kurtosis, and the time dependence. We did not, because we were limited by the Prisma scanner, we could not reach high enough B value to see deviation from linearity. So we don't consider kurtosis at all in this work. And we looked at the time dependence of the diffusion coefficient. And very luckily for metabolites that are only intracellular, the model is very simple. So you just have sticks with certain orientation. So yeah, this is also what Yana was asking, whether sticks is valid, but this is our kernel. And you can relate for this very simple model the mean diffusivity that you extract from your diffusion tensor to the DI, which is the intrastick diffusivity. And this is independent of the way the sticks are oriented, so independent of what we found before the ODF. So this equation is always valid for sticks, one type of sticks. So this is the case for metabolites. So what we did experimentally was measure the tensor, the diffusion tensor. We extract MD at different diffusion time, and we relate this MD just by a factor 3 to intrastick diffusivity. And intrastick diffusivity time dependence is something that is really interesting because it tells about structural disorders. So this is also something from theory from Dmitry Novikov that says that if you look at the time dependence of this intrastick diffusivity, you will access information about structural disorder, which means how non-stick the stick is. So all these statistical features that you have, for example, beading along the stick and non-homogeneous deformation of the stick, you will access this looking at time dependence of the diffusion coefficient, and this is what we call structural disorder. We assume in this work that structural disorder was in 1D, so this is just because we don't have enough experimental point to fit this exponent. So this can be 1D, 2D, or 3D. So we would need more to fit that, but we assumed here it was just 1D. So we have this formula for time dependence. And I was showing you before this plot of how the diffusion coefficient with diffusion time decreases and then plateaus to an infinite value. We are here now studying how this part of the curve, so which is to say how the D with time, how does the D changes with time as a function of square root of T. So we are looking for these two coefficients that will tell us about this non-homogeneous structure along the sticks. And experimentally, what we did in this part was we acquired the diffusion tensor, so like multi-direction, 6-direction diffusion tensor as a function of diffusion time. And we look at MD and FA as a function of diffusion time. So this is the experimental protocol, quite similar to the one before. We have now 10 volunteers for this project, same acquisition time, same voxel position. So you see here the statistics of voxel white matter is really dominant, so 94% white matter. So it's really almost pure white matter. We use the same sequence from Andre, but now we use oscillating gradients because we need to reach short enough diffusion time to probe time dependence. So we use 4 diffusion times from 12 milliseconds to 103 milliseconds, so 2 points with OGSC and 2 points with PGSC. And we have just one shell for this time. We have just this 1.4 shell, which is the highest we can achieve on Prisma, with 6 directions. So we really get the tensor with minimal, the minimum necessary acquisition. And processing is done in the same way. Also, we don't have macromolecules because T is even longer here. And diffusion MRI is done in the exact same fashion, same protocol, and we also have this very complex acquisition that gives us access to DKI and standard model. This is my second MRI slide. So the spectral quality that you see here is not the best you've seen probably. So this is an example of the B in blue here at the back, the B0, and then the 6 direction at B1.4. At the shortest diffusion time, you see here quantification, so really flat in the macromolecular region. And again, we quantify only the 3 major metabolites: total Na, total creatine, and total choline. Okay, so the first result that we were happy to see time dependence. So this is not something that is trivial. So we see that when we look at this is the diffusion coefficient of different metabolites as a function of time. So we indeed see that the diffusion coefficient decays with diffusion time, which is expected, but it's nonetheless hard to measure. So this is what we see here for all metabolites and also for water. About this, I will come back to this later to tell you what this means about the microstructure. Then we looked at FA, again, FA as a function of diffusion time for all metabolites and water. Here, FA is a bit more noisy to extract than MD from the tensor. Our acquisition is maybe suboptimal to extract FA with high precision. So, but we still see that there is a good agreement between metabolites and FA. So FA from diffusion MRI from water is found. You see here is the orange box and the gray box. So overall, you have a good agreement and relatively flat FA as a function of time, and a good agreement between metabolite and water. And this is also what we saw in part 1, that FA, with this B value protocol, FA was very similar between water and metabolite. And now going back to the diffusion time dependence. So this is the important results here. There has been a recent publication from our group which explains how these 2 diffusion parameters, so di infinite and CD, are related to geometric parameters. So the main goal is not just to measure diffusion, but to really understand what it means about the axon shape. And there is this series saying that these 2 parameters explain something about the beading. So the beading is how the axon cross-section varies along the stick. So this is just a normal phenomenon you have in healthy brain, but also in pathology. You just don't have like stick-like axons, but you have really a variation of the diameter of the axon along the track. And this is a way of measuring it. So you have 2 parameters that relate to this CD and D infinity. So this is a bit convoluted part, but the first parameter we look at is tortuosity. So it's D0 over D infinity, and it really says about locally how the AX, which is the cross section of the axon, how it varies along the segment of the axon. And the second parameter, which is gamma 0, so this one is a bit less intuitive to understand. So it's not about the local fluctuation, but it's about the large scale fluctuation that would survive at so a small Q limit, so at a large distance. So I give you an example here. This is from this paper. So you have different sticks here that have very different local properties, but still they will have the same gamma 0, which is the plateau here of spectral density function and at low Q. So these sticks, although they are very different local geometry, they preserve some large-scale statistics that remain the same. So we have the tortuosity that is more local, about really fluctuation of diameter, and you have the large-scale gamma 0, which is more about large-scale statistics. So we looked at these parameters for our metabolites and for water. So this is just first the diffusion parameter. So you see CD, which is coefficient here, and the D infinity. You see that the D infinity for water is much higher than for metabolites, as expected, because water diffuses much faster. And then we have the geometric parameters. So if we just convert these two parameters into these two new parameters, we have a new 2D plot. And here, interestingly, so water, which had much higher diffusion before, now merges into the same overall group, because these are really about geometry. It's not anymore about how fast water diffuses. So water senses similar structure. So it makes sense that these points all coalesce in the same plot. And so what we were happy to see here, but this is still preliminary. So if we look at this family here, so you have the water, which is red, the NAA, which is blue here, and we also added work from rat electron microscopy, which could also derive this two feature. We could see that this family may represent the neuron family, so that is probed by a large volume fraction for water, just NAA being mostly neuron, and for this EM data. So you can see that, yeah, there seems to be a family here that might be different from the family here, which is the family of total chlorine and total creatine. So if these results are confirmed, it might mean that we are probing two geometric families of sticks, which means that we would have distinct kernels for neuron and glial cells. So this was really also the main result of this part 2, that we seem to see that on this geometric plot, 2D geometric plot, we can separate what seems to be linked to neurons, which are Na, described by Na and water, and what seems to be linked to more glial markers. Okay, so now I'll go to the conclusions. Maybe I can do the conclusion and then you can ask questions on this part. So overall conclusions of these first two parts is that we can probe the glial compartment with diffusion MRIs, which was hardly accessible with diffusion MRI. We evidence tentatively, because it still needs more work to be confirmed, but different ODF from part 1 and different kernel for neurons and glial cells, which is measured in vivo. And this has several implications. So from the technical standpoint, we may wonder if the hypothesis of a unique kernel in diffusion MRI is valid, and if diffusion MRI protocols are sensitive or not to this additional kernel. And more on the biology side, it gives a unique characterization of glial cells, micro and mesostructure. So what I call mesostructure is really the ODF. And it will allow us in terms of to look at pathologies where glial cells are specifically impaired, and to try to see if we can measure this in vivo. We have several limitations about this current work. So we are comparing all the time, especially in the ODF part, the NA versus choline, but choline has a lower intrinsic SNR than NA. So, and because ODF is very sensitive to noise, it's always difficult to make sure we compare things with the same accuracy. And so this is still a work in progress. The cell specificity of metabolites has been disputed. So it's really a major hypothesis for all this diffusion MRIs work, and still disputed. We are limited by current Gmax of our scatter, which doesn't allow us to probe more than the DTI regime. We have small sample size, and we have to work at long TE, especially because specifically because we are limited by Gmax. Okay, so this was the conclusions of these two parts. Just my last very short part will be about the connectome scanner, so the new scanner we have. So if you have any questions here, I'm happy to take them.
One which is common to both parts anyway. You always say your SNR is not good enough for anything else than your 3 singlets.
Yeah.
But the SNR for the glutamate part at 2.4, 2.2 doesn't look that bad. So Is it really worse that camera for GLX, for example, compared to Grossman?
Yeah.
I remember we tried it, but I don't remember what the conclusion was whether we tried it.
Yeah, here it's quite so 20% compared to 2 to 5%. So it's much worse. But I think my main concern was also that glutamate doesn't tell me much about cell specificity. So I would want maybe minoxidil there. It would be nice to have a backup for choline for glial cell. Glutamate, I'm not sure. And plus glutamate.
But we have GLX,
yeah,
then it's complicated.
Yeah. So
minoxidil is how much? 20%.
Sorry?
20%.
Yeah. Yeah, so for now the T is really probably limiting the. So I thought it was convenient to have a long T because you don't need macromolecule in the base set. But yeah.
250 is a lot now. So
yeah.
Maybe you can go at 100.
The problem is we need the long T to be able to accommodate the oscillating gradients. So yeah, if you want to reach short diffusion time with this Gmax, with this is the minimum we can do. So with
On a prism.
On a prism.
Going to max, you might do PGSE at short T. How short you can get for
short T, yeah. That's
how short can you get on the connector max to reach?
I don't know. That's January work. Yeah.
Malte has it.
Yeah, Malte.
Yeah, hi Jessie. Thanks for the great talk. It's very intriguing. Especially like I was wondering about this two sort of stick populations that you find for the kernels. I wanted to ask you about your interpretation so it seems that the total creatine looks more similar to the total choline, so meaning that it would behave in a way more glial-like. But then if you think, or I don't know, my interpretation of the total creatine would always be, okay, it's this mixture between neurons and glia. And if you have like overall more neurons, I was a bit puzzled. Shouldn't it like then look more like the neuronal kernel, or?
Yeah, that's so I think there are several aspects here. So first, so you see the error bars are quite high. So yeah, the conclusion that there are two families is a bit far-fetched, but you can also argue that the family of purple points are in between if you look at the diagonal, are in between the family of neurons and the choline. So in that sense, we can argue that if creatine is a mixture of neuron and glia, it's probably what we see here. But yeah, I think this question of where is creatine and how many glial cells do we have in my voxel? What fraction of the volume, and if creatine is not in all types of glial cells? So I think it's very hard to conclude on total creatine. I think it's still I would be really interested to know what is exactly the composition of my voxel based on maybe Atlas work or something like this, to really say what type of glial cells we have, and do we expect creatine in high concentration in these glial cells or no? That's really an open question. Yeah.
Cool. Thanks.
Thank you. Okay.
How relevant would be to try it on another type of scanner where you could do this better?
Yeah, that's my part 3. Thank you. Yes. So this last few slides are about this new scanner that we received just in November. So it's really fresh results. This Connectum scanner, I don't know if you've heard about it. So it's a 3T scanner designed by Siemens in a partnership with MGH in Harvard. So they made this Connectum, what we also call Connectum. Now the commercial version is Connectum X, with very strong gradients. So they have 500 millitesla per meter gradients and 600 tesla per meter per second slew rate. So it's really a very impressive scanner for human. You see how it is located here compared to my Prisma is here and here. So really we will boost enormously all the performance. And it's a head-only scanner, which is different from the Connectome one, which had the problem of it was a full body and had the problem of PNS. So here, because it's a head-only, you can use basically full capacity of the Gmax and slew rate because your PNS is made smaller.
And
so
is it less noisy compared to the classic one?
So I've been inside and it's really tolerable, even for an hour and a half. Yeah.
The 3D.
Yeah, it's really it's much quieter than 9.4.
Why you were inside?
Inside, yeah. So no, it's really the tricky part is that the coil is really because you have in the normal 3T you have the body coil that has excitation. Here you have the coil so you have the receive, and then just around you have the transmit, and it's really you cannot be claustrophobic because you are really packed in a small space. So yeah, that's but the noise is really tolerable. And what I'm wondering still whether the table vibrates due to strong gradients. That's the experience of Andre as well for Connectome 1. I think they are trying to mitigate this, but yeah, overall it's not uncomfortable.
Sorry, you have said another point for the impulse gradients. Is that the Schema-X? Whereas you have this really high slew rates of 900.
Yeah, I'm not sure. That's the impulse is this CIBM? I don't know. What's the impulse is the
1070. But we can run it with 900.
yes.
Okay. Good to know.
Yeah. Okay, so I'll show you now the first scan. So it's really just diffusion MRI, and no MRS anymore. We acquired with the product sequence, so we are also limited in terms of performance to approximately Connectome 1 performance. But we just received the advanced diffusion MRI sequence, so we will be trying to test more the system in January. So the first thing we tried was high resolution diffusion MRI. So this can be done on Prisma, but not as fast. So we compared the 2 mm isotropic DTI protocol to 1 mm. So this is usually done. This is what is standard 2 mm isotropic in general. So here, when we look at we could reach 1 mm in a reasonably short time. So this was really interesting because you can also do that on Prisma, but yeah, longer acquisition time, also longer TE. So, and when we asked our radiologist to look at the images, he found all new regions that he could identify, and he was very excited to be able to delineate this part of the anatomy that I'm not able to explain you about, but overall we will be able to do very high resolution diffusion MRI. The second thing was benefiting from the high slew rate. We can do a high bandwidth for EPI readouts for diffusion MRI, which allow us to do this almost distortion-free diffusion MRI. So you see here the overlay. So the gray is the MP range at the back, and then overlaid with the FA map on top. And you see here with the low bandwidth, you have a distortion. So the corpus callosum doesn't really overlay with the corpus callosum on the anatomical image, which is a bit more at the back here. And this was the bandwidth we would use on, for example, Prisma. It's typical bandwidth. But if you go to very high bandwidth, as we can do now on the Connectome, you see that almost no distortion of the shape of the corpus callosum with EPI. So it's really, we'll be able to, so this is before processing. So we usually correct for these distortions, but now you can acquire natural data with raw data without distortion. So this is a second interest. The third interest has been very popular is axon radius mapping. So you can, because you can go to very high B value, you can tentatively measure the diameter of an axon with diffusion MRI. So this has been done before on the Connectome 1, and you see here. So you have if you look at the spherical mean, you can fit this function and then retrieve the axon radius. So we went here to be 16,000, and we could reproduce the DA perpendicular maps that were already published in literature with connectome 1, so it's the same scale, you see a similar contrast. And when we try to do axon mapping, so this is a direct conversion from the DA to the axon radius, you see here the values that we found. So it's just a sampled value in one of this regions. So you have a relatively realistic axon radius. They are probably too large, sorry, because the B value is not high enough yet. So this was a known problem that's seen on the difference between connectome 1 and connectome 2. You need very high B value to probe the realistically small axon radius. But interestingly, we could see that you expect a difference in radius across the corpus callosum. So you expect a higher radius in the splenium part than the genu. So this is also what we seem to see here. But this is really preliminary results that we got just in December. And now if I go back to my conclusion, I can add that because we got this very new scanner, we will be able to really extend the possibilities of our experimental protocols. We would like to try to go to shorter T and obviously higher B value to be able to extract something else than the diffusion coefficient, and will also allow to improve, hopefully improve our kernel and ODF feature extraction. Okay, and with this, I would like to thank everyone that helped me now, before, and my current group at NYU, Cristina for invitation and for everything you taught me before your group, and Andre Doring for helping with providing the sequence and being a great collaborator here on this project, also with Roland. And yes, thank you everyone for your attention.